I will introduce you briefly. Okay. All right. So good morning, everybody, or good afternoon if you are in another part of the globe, like our friend Peter. It is my great pleasure to introduce Professor Richard Lockshin, one of the greatest pioneers of the field of cell death. He actually coined the term programmed cell death in 1963 in his PhD thesis, which dealt with the hormonal control of silk moth metamorphosis. Together with his PhD supervisor, Carol Williams, he then published his landmark paper entitled Programmed Cell Death, Endocrine Potentiation of the Breakdown of the Intersegmental Muscles of Silk Moth in 1964, which for me is very touching because this was the year that I was born. So I was born together with the term programmed cell death. That's pretty cool, okay? And then he published a series of follow-up papers describing the cytology, the neuronal control, the influence of drugs and cytolytic enzymes that control the breakdown of the intersegmental muscle of the silk moth. So from now on, everyone, every time you hear again the term programmed cell death, you will remember Professor Lockshin and his talk today, okay, in his course here. So Professor Lockshin obtained his bachelor and PhD degrees at Harvard University, and after postdoctoral training at the University of Edinburgh, he returned to the USA to take up a position as assistant professor at the University of Rochester, where he remained for 10 years before moving to St. John's University, where he became a full and then an emeritus professor of biology. So he published over 100 papers, many chapter books, was a keystone speaker at many international and national meetings, conferences. He has received so many honors and awards over the years that I'm not going to be mentioning all of them. But what I would like to mention is his long life collaboration with his wife and dear friend Zara Zahedi, with whom he's co-authored papers and books and founded the International Cell Death Society that has been instrumental in disseminating research and clinical information on cell death. So Rick, thank you so much for accepting our invitation. And we are all very excited to hear from you. I think you presenting the viewers presenting, not the view itself.
Thank you for the introduction. It really is a pleasure to join you. And it's gave me a chance to reflect on some of the things. And one of the things that I would point out and why it's a particular pleasure to come or to do this is that it's been a long time theme of mine, and actually mine and Zara's, that science should be really an international enterprise. So a few years ago, when I gave a talk in Mexico, and I was reflecting that monarchs migrate back and forth, I introduced the talk as with this comment, which we were discussing a group that we have that we call Scientists Without Borders, that science should be like a monarch butterfly. It should fly freely from country to country, tasting the sweetness of every land and sharing its beauty with all. So this is for you in Portuguese, or in Brazilian. I won't really try to read it for you, but I think the sentiment is there.
So beautiful.
Beautiful.
Professor, yes, you can, how can I say, change your presentation mode there, because now you are sharing as a presenter,
So,
and
so,
you can,
uh,
could you
uh,
change
sorry,
your presentation
Display
mode? Yes, you
sorry,
can, yeah, that's
um,
it. Yes,
let me,
in the bottom below.
let me, uh,
Eu acho que ele pode
So
apresentar de
just
novo.
click in this.
Lá na versão de apresentador tem assim, Display Settings, ele tem que clicar lá e apertar Swap Display. Ele pode apresentar inteiro, full screen agora.
Não, mas agora é só ele clicar embaixo aqui, ó.
okay.
Richard, just click in the presentation mode there in the PowerPoint.
Sorry,
Okay.
what, is there a problem?
No, when you present, Rick, the first time it was all right, but now you are presenting the presenter viewer.
Oh,
Okay.
okay, you see that as well.
Yeah, I am
Okay.
seeing that as well. Okay, so now, yeah.
Okay, okay, so we'll go on from that.
So
Okay,
you
okay,
should
so we go on.
you
So,
should
this okay?
no, you should start again. Now we are not seeing anything.
Okay, okay.
Just
So-
just share again. No, that's fine.
Oh, I'm sorry, remember, sure, just a second. Okay. Okay. Okay.
Yes, perfect.
Okay, now we're
Yeah, perfect.
we'll
Thank
go
you.
on from there. So I'd like to set a theme, which I sometimes make the comment that science is an onion. So what do I mean by that? What I mean is the questions have actually always been the same. And what we do is that we keep just peeling off one layer after another. So we start with phenomenon. We ask what actually happens, when does it happen, under what circumstances. If we know the circumstances, can we control the phenomenon? How? What makes it up? How does the control actually work? What's the cellular mechanism of it? What's the biochemical mechanism? What's the molecular mechanism? Can we control the gene activity? Can we understand what the interactions of the molecules are? So these questions actually have been going on in what we're talking about today since the mid-19th century, since histologists first started seeing it. And now we just keep getting deeper and deeper into the question. So by the mid-19th century, when histologists took advantage of the coal dyes and the clothing dyes that made up the garments of the period, dead cells were seen and commented on. And so these were some of the images that people developed, generated during that time. And they say what we clearly recognize today as apoptotic type cells. However, that was a phenomenon. They, excuse me, they didn't know much about how it occurred and why it occurred. So in the 19th century, that was pretty much the issue, that you could see dead cells at various times. By the early 20th century, Aly Metchnikoff was studying phagocytosis and realized that cells died in that process. And shortly thereafter, Damon Fell, investigating collagen, realized that chondrocytes that formed the collagen died within that matrix. And so people were aware that these things were occurring, and it started to bring up the issue of what the causality of it all is, uh, which is um, uh, which is the second layer of the onion. So from that time on, people started noticing that in the maturing thymus, the thymocytes all died. In gonadal differentiation, uh, uh, embryos started with male and female organs, and in each sex, one set degenerated. And by 1951, Glucksman published a long paper describing the very different types of cell death that were known. However, he did this tabulation based on what he presumed to be the purpose of the death, which doesn't serve our purposes today very much, since we would rather know more about how it actually works. By the 1950s, Rita Levi-Montalcini, working in the laboratory of Victor Hamburger, realized that neurons could die and traced the factor to ultimately to nerve growth factor, which she finally invested her research in. So this is Rita Levi-Montalcini and Victor Hamburger. In the thymocytes, various scientists realized that glucocorticoids were a major um, a major factor in why they died. And in chick embryos, John Saunders pointed out that in certain regions of developing chick embryo, cells died on schedule. If he explanted them prior to that time, they would still die in culture. But if he transplanted them elsewhere on the embryo, the death was still reversible. So that started creating the sense that there was a control over the process of how cells died. That brings us to the 1960s, and it's worth noting what the state of the laboratory at that period. It had this sort of elegant appearance, and I will note one thing. This was sort of the source of distilled water, these jugs. However, this is a bit of a misrepresentation because for in our laboratory, the jug was a 25-liter jug, and we tended to put it up on shelves about this high. So it was somewhat, you had to be young and stronger than I am now to be able to do that. And it was always a little bit precarious of a process. During that period, we used glass pipettes, which we manipulated by mouth sucking and blowing, and we cleaned them in a horrible solution called chromic acid, which was a mixture of sulfuric acid and potassium dichromate, which we had to wash an infinite number of times to get clean. Our micropipettes were things like this. This was a big one, but it was called a Carlsberg pipette, and we could get them as small as 10 microliters. And the trick of them was that there was a constriction in them so that when you drew fluid into them, the surface tension would block the uptake to here, and you would get a fairly accurate measurement. And as I said, they were down to about 10 microliters. Another thing, this was our spectrophotometer, which looks not nearly as sleek as they are today, but it was really quite serviceable. But one thing about it was that it was run by vacuum tubes, not transistors. And so it was analog, which meant that you had to recalibrate it every time you used it. That was actually fairly good training because uh, we learned about calibration. And then that is things like this, in which I can really startle students today by asking if they've calibrated their system, particularly when they would bring me something, some measurements of fluorescence or uh, optical density or something of that order, because quite frequently at the lowest levels, um, uh, there's plenty of interference from other materials, and fairly quickly you saturate the system. And so, for various reasons, either electronic or because you use up the substrate, so there's only a fairly narrow linear region. And when students would present me data that they would be extrapolating this up to concentrations of this order, and it would be in the nonlinear linear portion, I would have to point out to the students that they were not correct in their measurements. But that was one of the things that we actually did. If we wanted to find a book, this was our search engine. I don't know if you've even seen these today. Our version of PubMed, the citation index, and Google was something like this: chemical abstracts or bio abstracts. What these did, they came out in maybe 100 page supplements every month. And in those, what was happened was that the titles of articles would be indexed by um, the words in the titles. So you would hunt for something, say, we would show the words around that thing. So you would look for something if you wanted to find, say, something about cell death. You might find this kind of article, and otherwise you could search for words surrounding cell. This was all done manually by teams. That must have been an exhausting and very boring amount of work. That's how we searched through the literature at that time. In the 1960s, there was no real genetics. At best, you could hope for Drosophila or Neurospora. During the period that I was a student, Crick first suggested that the codons would be 3 letters in length. There was the first suggestion that there was a messenger RNA. And before I graduated, the first codon was introduced. It was a somewhat marvelous story that would be great to tell at some other point. There was no Xerox machine. My doctoral thesis was literally on carbon paper. Xerox had just come out, but nobody was sure that it would be permanent. There were no acrylamide gels, no gene sequences. Radioisotopes were available, but extraordinarily expensive, and there were no. There was a scintillation counter, but no real ability to use them. There were no fluorescent substrates or labels for anything. Protein measurement was by the relatively inaccurate and easily insensitive and contaminated by your red reaction. There was no CRISPR. There were no DNA ligases. There was nothing of that sort. So what we can do today we couldn't even imagine back then. Today, we're at least 1 billion times more sensitive than we were in 1963 when I finished my doctoral thesis, and it still impresses me. We sometimes invented tools to get an intermittent stimulator, which I wanted, and which was available on the market, but not something that my advisor would invest in. But we had a stockroom guy who stored everything, and there he stored a clock which left horizontally. You could put mercury droplets at various locations on the clock, and you could connect it to a transformer, and which would wire to the center part, and one of the hands touching the mercury droplet would complete the circuit, and which you could then send to the animal to stimulate it every minute or every 5 minutes or whatever you wanted. So we got an intermittent stimulator by that. To measure minute amounts of proteases, you could take photographic film, which is basically exposed and developed photographic film, was silver granules embedded into gelatin. And so you could put a droplet onto this, and if there was a protease that could digest gelatin, it would eat a little hole into the film, and you could record that. So it was a pretty sort of try anything approach to life. At the time, cell death was considered to be what we know. It was all considered to be necrosis, where a normal cell would basically lose control of its energy and ion pumps, and what would happen then? It presumably would collect lactic acid and undergo osmotic lysis, and it would rupture and release the cell's contents and often create an inflammation. So it would look something like this, in which you would get uh, just the rupture and complete dissolution of the cell. Sometimes it would show a lot of blebs of essentially fluid-filled vacuoles before it would die. But there was a growing suspicion, because not all deaths looked necrotic in that sense. I showed you some of the early microscopy. And another thing was that many deaths occurred as part of development or homeostasis. So it seemed to be far more than this sort of uncontrolled idea that was considered to be necrosis. So now we got to programmed cell death. Basically, my mentor, Carol Williams, suggested several ideas to me for a thesis. He was a famous insect physiologist, and one of them included the giant silk moths of the Americas, in which to inflate their wings, when they come out of the cocoon, they have a series of muscles in the abdomen that contract quite vigorously to pump fluid into those wings. However, these moths never actually eat or drink, and this represents substantial reserve of energy for them in the few days that they have to live. So within 2 days after they emerge, all of these muscles deteriorate and are gone. And so he suggested to me that people know about this, but they didn't know how it actually operated. And one of the things that I knew, having an undergraduate degree in biochemistry was that Christian de Duve had recently described lysosomes, which were, and that this might be one of the ways we could look at it. So what we did in those studies, we had available to us physiology and crude biochemistry. That's what we could do. What we found was that the death of the muscles followed a sequence. We could identify an endocrine potentiation, we could identify a neural trigger, and we could identify activation of lysosomes, appearance of lysosomes in the tissue. That sort of led us to a hypothesis that if a death can be predicted in time or place, it's inherently a genetic event like any other, and that a sequence of events leads to the destruction of cells or tissues. And this was sort of, I certainly was aware of, and we were comparing it to, for instance, the death of gonads to determine sex, the death of the thymocytes, and the disappearance of, say, a tadpole's tail. So programmed cell death at that time described a sequence of events and implied that any biological event that could be predicted was inherently genetic. But attitudes change, and so now it tends to refer to any structured process leading to the death of cells. Meanwhile, people were getting more worried about necrosis. Around 1967, John Kerr described, he was a pathologist in Australia, described what he called as shrinkage necrosis, that instead of swelling and bursting, cells shrank and the nuclei collapsed. It was a fairly spectacularly different from necrosis. He described it then in 1967, and in 1972, he was on sabbatical in the laboratory of Alistair Curry, and Andrew Wiley was a graduate student there. And they introduced the term apoptosis. But they chose it, it meant the falling of leaves in the fall, or falling of hair as one gets balder. But a Greek scholar has suggested it, and particularly it implied that apoptosis completed the cycle begun by mitosis. This is what it looked like and what they published. The cells shrank, the organelles became very indistinct. The DNA condensed into these blobs, really, and the cells fragmented and blebbed. And so that was what they were calling apoptosis. It's worth taking an aside here and making a note about semantics, and it's in defense of original writing, as opposed to letting yourself get drawn into, say, chatbot writing for you, and in defense of you're using your own voice. And that is, I had considered that what I was observing was basically obvious, but in choosing the terms programmed cell death, it was stating the obvious, but it was catchy and modern. People were just beginning to talk about computers at the time and what computer programs were, and it was a sort of cool word to describe it. Carol Williams was always very much in favor of cool descriptions, and we were always trying to emulate them. Likewise, apoptosis described an observation and declared its generality. But the illusion of mitosis captured the imagination and the word caught on. So the choice of words is important. I think if I had just said death of metamorphosing insect muscles, it would not have gotten noticed. And if Kerr had stuck with shrinkage necrosis, it would have gotten far less attention. So again, your own voice and choosing words to make points. Ultimately, though, you really have no control over the evolution of meaning. So, yeah, so things get argued over and meanings change. So those two developments built enough energy that in 1981, we published the first book on cell death in biology and pathology. Again, the title is far more boring than I would have liked. I wanted to just call it cell death, but people asked me, was it going to be about violence in prisons, or was it going to be about arms sales? So we finally settled on the more boring one. But it captured many of the themes that were being developed and anticipated the upcoming role of nucleic acids in the field. So now we move into what I would consider the more modern period. During the first period, there were many efforts to constrain everything to apoptosis and to reject anything that did not fit that. And so meetings were filled with many arguments about what was or was not apoptosis. This is what I mean by when I say that words change and the public creates them. So during this time, we were gradually growing to the acceptance of an important process and the definition of variants of apoptosis. There were alternates, but though still organized, mechanisms by which a cell self-destructs. So, although the arguments were in fact very heated, it's worth noting that that a consensus was beginning to develop. Still, it was a fairly slow acceptance. My first paper came out in 1984. Wiley and Curry introduced the term apoptosis in 1972, and our book came out in 1981. But it was still a good 10 years or so before publications started to rise rapidly and really almost not quite exponentially, but there are now over 250,000 publications in the field. So the question is, what happened? What happened in those 10 years that suddenly made the story interesting? Well, the idea that there was a form of cell death that we could call autophagic cell death that didn't depend on what we're now getting and be known as characteristics of apoptosis, such as caspases and the involvement of lysosomes in the process. That idea was growing, and apoptosis was clearly distinguished from necrotic processes, so that there were clearly forms of cell death that did not cause inflammation and which allowed cells to die fairly peacefully. So Christian de Duve had first introduced the concept of lysosomes, which he called that because he was studying when he realized that they existed. His first tests were to try carbon tetrachloride poisoned liver. And he found that in that condition, lysosomes ruptured and leaked their enzymes into the cytoplasm. So he assumed that they were potentially the killers of cells. And then in terms of bringing it back to the cell death story, Zara Zakari started pushing for the fact that you could have a programmed cell death or a organized cell death without uh, without caspases. Maria Yahla started finding situations in which that cell death caused by autophagic processes were important in various disease situations. And Beth Levine, using yeast, started working out the genetics of how autophagy actually works. So that those developments were contributing a lot. Again, depending on the techniques that were rapidly becoming available during that period. Those techniques included molecular biology, which was now developing means for sequencing genes and controlling the sequence, and even beginning to alter or mutate those genes or to knock them out and to see what happens. The genetics of the roundworm Caenorhabditis were coming along quite rapidly. Immunologists were recognizing different types of cells that played a role in the immune system and identifying the potential antigens, and then technology was coming along to identify receptors on these cells and how they worked. And we were seeing the rise of fluorescence and other labeling techniques to be able to allow us to isolate and identify different types of cell death in situ. And that was important because, as histologists had pointed out, that the liver could disappear in within a month from homeostatic turnover, and you would still see only one or two dead cells in any given section of the liver. So the history of the research is I gave you a copy of a review that we did in 2001, in which it was following many of these early steps, and I would break it around the 1990s when this started becoming important. So what happened? There was the rapid development of new technologies, and that led to the rapid phase here. Cell death genes were first recognized in Caenorhabditis in 1979, and other techniques for observing cell death were coming along that period. Apoptosis genes started to be identified in starting around 1989. And so particularly the technology really started becoming fruitful around 1989. Also, techniques for labeling dying cells, such as the tunnel technique, which marks fragmented DNA, and phosphatidylserine, which marked a marker of a dying cell, came into being. So by that point, we're now getting a fairly good understanding how caspases actually work, or say how apoptosis actually works. And it was becoming apparent that in general, in the mammalian group, there was a caspase 3 which is a major protease that could damage the cells. And what was really getting to be interesting is that caspase 3 had a homologue, which was molecularly related to genes in sinusoiditis called cell death gene 3. The caspase 3 could be activated by in a couple of ways, which included other related caspases, and they could be controlled by adapters uh, 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 interacting with various regulators and effectors, so they could be inhibited or activated by a fairly complex sequence. What was really important here was that these sequences were conserved from worm to mammal, and they not only the form of the sequence, but the types of molecules that actually did it. So for that, we can thank Bob Horvitz, Judying Yang, who first sequenced caspase 3, and realized that it was related to mammalian proteases that were known but not known to be involved in cell death, and Michael Hengartner, who sequenced caspase 9 and found its relationship to the others. So there was this gradual improvement in techniques. And probably most important was the realization that that these processes were relevant to diseases. During this period, Jean-Claude Ameisen raised the question that AIDS might be an issue of how virus-infected cells die and how one might protect them against death. Then Luc Montagnier isolated the HIV virus that causes AIDS, and Marie-Lise Goujon continued the study of how the AIDS virus can kill cells. In cancer, there were many rapid developments. One of them was that there's a cancer called B-cell lymphoma, which was recognized as being not a condition of overproliferation of cells, but of failures for the B cells to die on schedule after they had been produced. And that led to the analysis of a gene that could do that, which is called BCL2, and which David Vo pointed out that this mutation was the cause of the cancer. Then Stan Korsmeyer and many others analyzed how it actually worked. There was also P53, which was known as a gene that was mutated in many types of cancers. Bert Vogelstein had pointed that out. And analysis of what by several groups, and including Scott Lowe in the United States, established that P53 worked because its normal role was to kill abnormal cells, meaning frequently cancerous cells, and its mutation allowed those cells to live. And then in the immune system, Pierre Goldstein pointed out that not only that killing of cells in the immune system was not simply an attack by killer cells, but happen by the self-destruction of the cell, starting with a cell surface receptor and Shige Nagata and Peter Kramer and Xiaodong Wang elaborated how that could be. So these were three major medical areas that and that became relevant to the cell death story, or the cell death story became relevant to them. So the consequences of all of this followed fairly quickly. Sorry, first of all, uh, government funding, like agencies like the NIH, through several sections, started funding studies of cell death. They started getting meetings on the subject. Commercial organizations such as Genentech and others started investing in it. And uh, medical schools and other faculties started recruiting specialists in the field. And it began the exploration of many other areas, including Alzheimer's and many neurological diseases, birth defects, developmental defects, autoimmune disorders, and suddenly the role of cell and even heart attacks and strokes. So the subject became significant to a lot of people. So momentum was building. The first meeting on cell death was in Sardinia in 1989, and another one in Europe called a recent meeting 1990. And there was a Banbury conference at Cold Spring Harbor in 1990. The Sardinia meeting was in honor of Emanuel Farber, one of the world's great pathologists. He was the one who named the stages of cancer. He pointed out that many mutagens form DNA adducts, and a major uh, industrialist and politician named Diana contributed money to create the Dana-Farber Cancer Research Institute at Harvard, honoring Emanuel Farber. However, so this was just a memory of this. If you can identify anyone in here, good luck. Emanuel Farber didn't really believe in apoptosis, and so he made the comments at that meeting and published shortly afterwards these comments. And I'll highlight them. He said, apoptosis is considered by some, especially by non-pathologists. I take that to be fairly sarcastic comment. And so he says, there's nothing, no field of basic cell biology and cell pathology is more confusing and more unintelligible than the area of apoptosis versus necrosis. He did have a point there, and because a lot of people were arguing it, and I think we have a more generic overview today, and it makes a lot more sense than at that point. So going on, Cold Spring Harbor series started in 1995. The Gordon Conference series started in 1995, and the Keystone Conference series started in 1998. This is a picture from the at Banbury conference. You can do many things with this. You can admire the elegance of the scientists at the time, discuss the dress codes at the time, or have fun trying to identify some of the scientists and then and particularly those whose names are still known in the field. But it was the start of this period. So societies were founded in the International Cell Death Society in 1994 and the European Cell Death Organization in 1995. This our first Cell Death Society meeting was in started at Rockefeller and then moved to Queens College, and it has been going ever since. So there was a growing insight into why cells die and a growing interest in that. So we learned the difference between intrinsic versus extrinsic apoptosis and the role of mitochondria and cytochrome C in it. So in a brief description here, you'll hear a lot more about it in the course. There was the cell death receptor pathway, and that is where a receptor in a cell's membrane picks up a ligand delivered for one of several reasons, like so there's fast terminal necrosis factor, trail, several of those. Those interact to release and activate a procaspase from the membrane, which then activates the killer caspase 3 or 6 or 7. Then there was the mitochondrial pathway or the intrinsic pathway, presumably being triggered by some metabolic events, but involving several inhibitors and activators in the field, releases cytochrome C to the cytoplasm, which together with another procaspase joins into an apoptosome, which then activates the killer caspases. So this was the result of the very rapid growth of the field. There were many players involved in this. So from 2000 onward, there was a proliferation of names for different types of cell death variants, if you wish, of apoptosis. There was necroptosis, ferroptosis, pyroptosis, parthanatose, and others that you will be hearing about during the course of this class. And some of these will persist, some probably not. We had intrinsic apoptosis, extrinsic apoptosis, and mitochondrial permeability driven mitochondrial permeability transition driven apoptosis, which was for various reasons sometimes distinguished from intrinsic apoptosis. This comes from a review published in 2018. So that brings to me the question, where are we going? We have a lot of new microscopic techniques. This just came from a meeting, I came from a meeting in which some of these were being shown, super resolution, in which you can look at the interaction of molecules on specific receptors on specific parts of cells. There are many others allowing you to identify gene activity and protein activity with immune technology using antibodies and various labeling techniques. Computing skills are growing very rapidly. uh, artificial intelligence can uh, uh, can search among the known data for various correlations and statistical correlations that might imply logical or causative roles during that period. And big data, the huge amounts of information being produced by uh, 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 genetic analysis and heat maps and cross-species comparisons is really expanding our knowledge hugely. So think about it. We can now visualize single molecules interacting with specific parts of other molecules in specific locations of known organelles. To me, that's fantastic and something that we could have never imagined would occur in our lifetimes. There are dangers to that, however, and you need to reflect on it. It's easy to get overwhelmed by the data. I just came from a meeting in which people were putting up heat maps, sequence analysis of genes and reaction sequences that sometimes look like the bowls of spaghetti sort of splayed out on the table. Huge, huge, huge amounts of data. And you need to remember what you're doing. You need to remember to tell the story, to keep yourself. What is the point of all of this data? What is it getting to? Some of the speakers at the meeting did not. So, what does it mean? What are the connections between one point or another? What are the future possibilities in it? You need to step back periodically and take a look at that. One of the writing grant applications can be a very tedious occupation, but it does make you stop to think about what it all means. There are still questions that are wide open. Pathways are not unique. If a cell commits to die and you block one pathway, the cell is still sick and can find another pathway. For a while, people were uh, looking for money and grants and setting up programs trying to block apoptosis. But if you don't relieve what's the problem that's making the cells sick, it still dies. If it survives, it may go into quiescence, which may be useful, or it may undergo autophagy, which again will reduce it to an inactive cell. If it's a cell whose activity is important to the body, you still have not resolved the problem. You still have to keep in mind that even if you set up a culture, for instance, which you understand very well, and which you add something that will induce apoptosis, the cells will die at different times, and not all the cells will die. We really have no explanation for this at present. There may be historical, metabolic, or other reasons. Perhaps some cells have more nutrition than other cells. Perhaps some of them in their history have suffered otherwise imperceptible damage to their membranes that others have not. There may be many other reasons. We really do not fully understand why cells die. So to summarize it, there was a article that Jerry Molino published describing his view of the history of cell death. It's in Cell Death and Differentiation, in which he shows pretty much the sort of same things that I did, but he correlates it with also the appearance of the journal Cell Death and Differentiation, Cell Death and Disease, and Cell Death and Discovery. Um, uh, and I just point out to you here, it's the same story I indicated earlier, that he missed a couple of items. He points out the Nobel Prize to Bob Horvitz, to Osumi for autophagy. He missed Aly Mechnikov. He missed the Sardinia meeting starting at, and he missed the appearance of other journals such as Apoptosis. So just keep in mind, any one of us who writes a review has limitations and things that we forget. So in brief, most cell deaths are programmed in embryos, in turnover, and disease. If in the 1990s it became possible to easily measure cell death, it became obvious that several cancers are diseases of cell death, and several other diseases related to cell death, and that caspases are near universal enzymes that can kill cells. In the early 2000s, the ideas developed there are many different types of controlled cell death. Starting in more modern times, molecular gene manipulating super resolution techniques take over, and we're entering a whole new world. So, as reprise, science is an onion. The questions remain the same, but the answers change. So, you started cell death can be predicted. How? It occurs in steps. It's programmed. How? It differs from necrosis. How? It has a genetic basis. What is that genetic basis? There are genes for cell death. Which are they? What are they? Some genes are required for cell death, some block it. What do these genes actually do? There are protease inhibitors and activators. How do they work? They're activated externally, that is, one cell saying to another, I will kill you, or metabolically saying, I will commit suicide. What does that mean? There's a sequence of caspases. What is that and how did it evolve? They're activated externally or metabolically. By what mechanism? Some work through mitochondria. How does that work? Maybe they work through controlling the mitochondrial membrane permeability. How does that work? Some things bind to the mitochondrial membrane and change it. How does that work? It's still, science is an onion. You still keep plowing through it and finding more questions to ask. So why should you do it? Well, you're scientists because in a sense you're poets. When I was an undergraduate, I took a course with a well-known American poet, Archibald MacLeish, and he liked to compare scientists and poets. He said, and poets see what everyone has seen and help to see them to really see it for the first time. Scientists see what no one else has seen and help others to see it for themselves. But you're a scientist because you have an interest in the world. This is a letter I sent to students the day after World Health Organization declared COVID to be a pandemic in March 2020 says, this is why you are a scientist and this is what you're trained to do, meaning telling the students that it was their job to understand what was going on and to explain it to others who did not, and to try to prevent utter nonsense from arising. And pointing out your knowledge is your best and really your only weapon. And I quoted a proverb, for wisdom is better than rubies, and all things desirable are not to be compared to her. Again, we have a responsibility to do our science, to understand how the world works, and to communicate it to others. Remember also that it's for everybody, for all of us. American science grew because it is a peasant science. When I first entered college, the science was pretty much aristocratic and patrician. It was highly intellectual, and it was a clean hands science. You could tell this by the names of the scientists and the way they spoke and the way they spoke down to students. At the time, however, when there was a rising in the States, a new generation of scientists who were largely immigrants or the children of immigrants. And they brought to the field this sort of peasant approach. They brought joy, they brought dirty hands, they brought this, let's take it apart and see how it works mentality. It was this mentality of, gee, I can play in this mud pie all my life and get paid for it. And it was a great time to be alive. And you could tell this from the young scientists who are now becoming prominent and speaking. There are many stories of that. The other thing that becomes important is that every group has something original and creative to bring the field, and it's for you to identify what is original and creative to bring to the field. All you really need is curiosity and the willingness to plow ahead on it. So remember, every day is the best day to be in science. So many things come along every single day that that you really cannot leave it once you've gotten into it. So to wrap up, I want to thank Zara Zakari and Miriam Lockshin, who did a great deal to design this talk. And if you found it not to be my usual very boring presentation, you can thank them on it. And thank you. Muito obrigado. So I will stop there, and I don't know that if anyone was trying to. Are there a lot of chat questions? I wasn't seeing them. Let's see what.
Hey, Rick,
Thank
first
you.
of all, I can't thank you enough for this wonderful and inspiring presentation. It was excellent. And the fact that you allow us to broadcast and register will be also, a bonus for us to go back and then watch it again. It's really, really, really inspiring.
And to be transported into this story by someone who lived every part of it. You just remember me.
I'm not hearing.
I love science. It's incredible. Thank you.
Let me just look at some of these questions that were raised, and maybe I'll come back to others. When and why did you decide to focus on your research on developmental cell death? Well, basically, uh, when you start as a student, at least in the States, quite frequently, a mentor suggests several projects that are, or several ideas that are of interest to his lab, and you start playing with them, and you see what starts to pay off. And among the projects that started, uh, since uh, in Carol Williams' lab, I was the one that had the most biochemistry. And I started looking for proteases. That came up fairly quickly, and it just grew from there. And so uh, that just happens. Uh, Question about, can it be applied to other types of cell death, such as bacteria or fungi? Um, there's actually a lot of research along those lines. Jean-Claude Ameisen, before he basically became a moralist for the French scientific community, was interested in that. And a uh, there was a Banbury conference going probably to uh, a cell death, a Cold Spring Harbor series on cell death in fungi. People made a lot of effort to try to compare it uh, to apoptosis. There are a lot of differences in it, but obviously you still have the same issues of uh, a cell living within a society and uh, the roles of other cells influencing it and its role in studying it. So, see if I what did you find most difficult in studying cell death? Hmm. I wish uh, I could have adapted to the molecular parts uh, more rapidly. You do get sort of stuck in the generation that you're working on. Another thing that when the genetics started breaking, I was teaching physiology at medical school, and I already had too many people uh, finding uh, their work on insects completely inappropriate for a medical school. And if I had dared to try to suggest that a roundworm might be interesting, uh, I would have gotten such a round of boos that it wouldn't have worked. Uh, um, the Why did I choose the specific part of those muscles? The biggest advantage of it was that looking at places where you could see a lot of cell death, most of them, whether you talk about tadpoles or insects, occurred at times where there were, say, massive number of other changes. I mean, when a caterpillar turns into a pupa, virtually the whole caterpillar is destroyed. And physically, you have very little opportunity to manipulate it uh, without causing severe uh, severe damage. Uh, the muscles had the advantage that very little was occurring at that time, and the adult insect was much more stable. So one could hope to manipulate it more than others. And if you start to think people who want to experiment, say, with gonadal differentiation, would have to manipulate embryos. And even if you did that in chicks so that you could get access to it, you did a lot of damage to the embryo in doing anything. Uh, uh, I think based on experience, the perspectives and expectations for the future, I think I addressed that at the end of it. Talk. If you want to know more, let me know. Uh, uh, Apoptosis uh, is surprisingly well. This is for Igor Hussein's question. Is surprisingly widespread in the biological world, where there are serious differences. For instance, the paradigm for apoptosis was seen in thymocytes, which are cells that have very large nuclei, relatively small cytoplasm. And so the nuclear involution became a very important thing. When you're dealing with very large cells, such as muscle, such as mammary gland, or cells that have many complex functions, such as neurons, uh, then you see a lot of cytoplasmic changes, autophagy, and many other kinds of things. So that's what caused a lot of the confusion as people were trying to make everything a thymocyte and uh, were rejecting the other types of clearly still organized cell death. So, uh, 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 I yes, molecular biology revolution was absolutely crucial. Again, I said the questions were always there. We couldn't really uh, deal with them until it became possible to manipulate genes to understand the structure of genes and so forth. And everything is going to go there. Um, you think there are many other types of cell death than those ready to start? Um, I would tend to look at it less like like the color wheel that in Galuzzi's article of summary of the types of cell death, and more like uh, like a rainbow in which if you really look at the edges of each color, they actually just blend into each other, or expect a rainbow developed, say, by a prism or something like that, in which they blend to each other and they might vary within themselves. So, I mean, it's a much broader process. And I think the idea is much more understanding how things work. As I said, you can draw biochemical sequences and pathways that look like a, a bowl of spaghetti dumped on the floor, uh, and grasping those and understanding them is just a hugely demanding intellectual task. Um, I would point out to say there were some ideas that looked fairly hopeful. For instance, there was a group that was considering that in a heart attack or a stroke, um, the cells that are seriously injured downstream of the stroke take several hours to die. And if you could block apoptosis and using modern medical techniques, relieve the anoxia by breaking the clot or opening the vein again, the artery again, that blocking apoptosis might really serve a function as a delay tactic to prevent this cell from making its final commitment to die. Uh, Beyond that, I think we're going to have to deal with the huge interaction among these processes. Uh, uh, why do they have different mechanisms? So the different genes involved. Uh, I would have to pass that to the evolutionists. Again, I think evolution works with whatever works for the moment. And although the structural pathways are the same, for whatever reasons, insects develop different slightly different routes on the process. Plants have very different reasons for killing cells as they grow. They have very different physiology, so they exploit those kinds of things. Lucas asks, is autophagy a cell death mechanism or a cellular mechanism for homeostatic control of energy metabolism and macromolecule renewal? That sort of illustrates the argument of everything blending in everything. Cells can go on perfectly happily, replacing mitochondria that get damaged using autophagy and eliminating other materials that have accumulated in the cell or are toxic to the cell. And they can go on quite happily doing that. If part of the question with the muscles that I was looking at, that I was asking, is this a homeostatic mechanism that's waiting for the savior to arrive, and the savior never arrives? You know, that to destroy. What happens in the muscle is the myofibrils get slowly consumed, and that process starts before the mitochondria collapse and respiration fails. And you have to ask yourself, is one way to create a programmed cell death simply to turn off the savior or the resuscitating mechanisms? So I would say it's both and all of that. What we now, most of the fundamental questions in science have already been largely explored. Is it harder to make big breakthroughs in science? No. The fundamental questions in science have been defined, and the definition gets more and more precise. And I said it's still an issue that it's an onion that we're looking at, and we if you look at a cell and say, why does this cell die in this time and place, and the cell next to it does not die? That question is still open. But whether you're describing it as a philosophical point or whether there are still parts of the metabolism of the cell, it's getting more and more complex. And that you who are growing up in a world of big data and extremely advanced technology will be looking at those questions in a way that I never did. I mean, at one point in my career, when I was at University of Rochester, Hans Holzfreiter, who was one of the great early embryologists, was still there. And I was talking with him one day, and he was showing me a plate with a giant slime mold, and he was saying to me, he was looking at it and saying, we still don't understand the big questions about this slime mold. And I was thinking, such things as, how does it organize in a spatial sense, how it shall move, and how does it manipulate all of that cytoplasm? And he continues and says, like, does it go to bread or cheese? And that was sort of the old timers approach. And I suspect that's sort of my approach. But you will look at the world the way you see it in the way that you have grown up. I mean, one of the things about trying to build this talk is my daughter Miriam was pointing out to me that you cannot fill slides with tons of data anymore because people don't deal with it that way. So, what's the most complex type of cell death to study? Basically, I don't know. I would say that if you can work out all of the parameters of any type of cell death that you're studying, such as uh, what what is forcing the death, what is blocking the death, what triggers the whole process, what is the sequence. Uh, it's all very complex, and you'll be studying that. Um, what would I say? A single discovery is the um, well, a single discovery that would be largely for change, responsible for changing the view of what cell death is. I would say probably the recognition that Caenorhabditis and humans uh, uh, follow uh, uh, follow re, re, uh, evolutionarily related mechanisms. I would think that would be fundamental to realize that um, uh, I mean, there are many points that are one could pick up. Uh, I mean, certainly the recognition that it was medically important was a huge issue. And, uh, you know, I always hate to answer a question like this because I will always forget something that that others will consider to be very important, and which is uh, yes, current techniques like omics can certainly expand the knowledge of proteins participating in cell death pathways. We're just beginning to explore which molecules interact with which other ones, what uh, what conditions influence that, such as the ionic conditions in a cell, how much calcium is available, how much iron to deal with something like ferroptosis, uh, uh, whether sodium potassium ratio is hugely important, whether the extent to which the acidity of the cell plays a role, the extent to which protein interactions causes the changes of shapes of different molecules, including DNA, and the extent to which that affects their organization. Uh, Formulating the hypothesis. So what was the crucial question led you to answer this mechanism? I couldn't really say. I mean, I started working on it, and then as I think it just grew on me that as I saw one step after another, as the endocrine steps and the neural steps and the beginning development, the appearance of lysosomes before the cells actually died, um, it just occurred to me that it was very obviously that had to be biologically controlled. And as I said, it struck me as an obvious statement, and the choice of the words uh, was became important. Uh, 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 the studies involving AIDS, cancer, immune systems, and others evolved together. Uh, yeah, I think as the technology, basically techniques for identifying dying cells uh, grew fairly quickly. And as people started realizing they were dead cells in different situations, they were working independently, and uh, it all started turning into uh, sometimes out of meetings that you would have an immunologist talking about uh, the, for instance, the uh, disappearance of antibody producing cells after the infection has passed, or the destruction of potentially of in the thymus of cells that could potentially produce autoantigens, and people who are looking at cancer and saying, but P53 is a uh, 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 generally causes cells to activate the process. They would come together, and the meetings were very important as people, as immunologists, turn to a someone studying cancer to turn to somebody studying uh, neural cell deaths. So in the 1981 book, by that point, I made it. Basically, I was invited to join the book because the uh, the publishers were concerned that it at the time was looking fairly narrow. So I invited Andrew Wiley to it. I invited uh, Rita Levi-Montalcini to it, and a couple of other people who got known in the field, and we made it this much broader book. But basically, people were hearing through meetings and things of other people looking at the same thing. So that's where all this came from. Uh, I think any explored area in relation to cell death, I think I tried to cover what they were. If you want me to try to amplify it, I'll, as soon as I finish scrolling through these. Uh, uh, um, uh, Question is that there are no particularly effective cell death drugs. Uh, Basically, as I commented earlier, um, there were efforts to use drugs to block apoptosis and various sort of things. The issue is still the question uh, of the complexity of the event, and still dealing with the issue of why is this uh, uh, sick cell dying? And the question is, why is it sick? And can we uh, relieve the problems that are dealing with it? Or conversely, why how has this cancerous cell escaped being killed by surveillance mechanisms that have done it? I think the issue is that it's just very complex, and the early solutions that everyone was looking for, like blocking apoptosis, turn out to not be very functional. Uh, uh, I haven't made, the question is about other uh, uh, unicellular eukaryotes, eukaryotes. Actually, Jean-Claude Ameisen was raising those questions, and he was looking at them, and I'm trying to remember uh, the British scientist who was talking about cells as a society. The problem is that it's very hard uh, to identify cell death in a unicellular organism because it just disappears from the culture. So uh, I there presumably is a literature about that now, and I'm and you're thinking about land plants that also have tissues. Uh, so I don't see why it couldn't evolve further. And as I commented that land plants have very different needs and physiologies. So for instance, they can want to make vascular tubes, and they do so by killing cells. They often want to uh, to change their leaves in the temperate latitudes. They often want to shed their leaves on a seasonal basis. Uh, some plants, say like um, um, the words escaping me right now. The ones that uh, uh, form groves along uh, the edge of the ocean. Um, yeah, come to me. Anyway, they collect salt in leaves as a way of getting rid of excess salt, uh, and they drop them. Sure, there are many. Uh, uh, I don't see any problem with cells using mechanisms that they already have on board to or organisms using mechanisms that they already have on board to get rid of cells that are damaged or which serves another purpose for the organism. Uh, so they can presumably evolve techniques to assure that, but I don't think uh, uh, the evolution appearing more than once, I think is just branching on different uh, schemes. I don't know if that says anything. Uh, how carcinogenesis process and different steps changes the cell death through the cancer progression or cell death shapes the carcinogenesis. Um, uh, I think, I mean, I would agree to say all cells, including cancer cells, live in societies and they interact with those societies in whatever ways you want. And they can be very selfish about it and ignore pressures. I mean, anything from hormonal pressures to cell killing uh, uh, killer cell mechanisms. They can ignore those mechanisms and fight, or they can respond to them. And I think each cell is adapting to the circumstances that it finds with mechanisms that it finds, and the organism is asserting what mechanisms it can control to to control that. I mean, um, and you see circumstances in which, for instance, if there is a mutation in a sequence to synthesize a specific hormone, as such as the hormone is not produced, the gland that is trying to produce it keeps increasing in size to try to deal with that. And so with any more mechanisms and potentially new therapies, I think we're really getting to the issue of understanding what it is that takes a cell to the threshold of death and the meaning that that has for medicine and therapy and how we can deal with it in that phase. We think that small non-coding RNAs, such as microRNAs, play a role in cell death. I don't know if you know the name of Devrim Gozulichik from Turkey, who's working essentially on that question, and and he thinks it's very important. And what I would say is that as molecular biology developed, we we had a paper that went somewhat unnoticed by pointing out that there were new proteins synthesized just before cells die. And so we went to see if we could get the RNAs for them. And what turned out is we found a group of short messenger RNAs that were produced at that point, and they were too small to be coding for anything. So, so we had no idea what they were doing, and we didn't publish it. And for all I know now, they may have been microRNAs. But again, this is something that we'll be learning more and more about. Why was cell death originally poorly studied? First of all, you couldn't really see it, and in comparison with differentiation and growth. And I always think that attitudes were such that it was pretty much, in the Yankee slang, it was a downer. It was sort of depressing to think about that. I'm more interested in things growing, and it was harder to study. As I said, dying cells just disappeared, and we didn't really have mechanisms to prevent their death and see anything of that. So it just seemed that it was much more fun to see how limbs grew, and then and to try to grow them on something else. What was the biggest challenge I faced while studying researching cell death? I was going through life sort of doing my own thing, and I didn't expect to say, for instance, people in medical schools I considered it interesting biology, and I didn't really expect people in medical schools to be so uninterested in it. And then there was this period. I mean, I don't think I ever felt despondent. I'm not that kind of a person. But what I did, I do remember, is there were years in which there were arguments over the definitions of words. And at this meeting in Belgium that we just had, I introduced Maria Yatala, and in researching how I would introduce her, I was looking at papers and comments that she had made in her life. And uh, she remembered and described in one interview that a uh, and that the projector had died, so she was ad-libbing about her observations about autophagy, and a distinguished Swedish professor. I think I have an idea who he might have been, but I didn't ask. Got up and said, "There is no cell death without caspases." And uh, so she was at the time a young scientist, and she said she felt uh, very upset about that, but and was happy the projector came back to life again. But uh, 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 this argument was going on, and people could be really very vehement about it, and uh, it it really wasn't worth it. To some extent, uh, there was issues of pride, is who gets to claim the name of something new. You know, that's sort of nice. To some extent, there was this sort of uh, digitizing of the information. There had to be this one pathway, and that had to be distinct and special, and then another pathway that had to be distinct and special, and another one that nobody was interested in, and so forth. And those uh, that desire to keep everything as binary or digital uh, created a lot of fury and uh, uh, and frustration during that time. After that, uh, there were times in say when people weren't interested, and that was reflected in interest in a grant application or something like that. So there were times then, say, a grant reviewer would just sort of dismiss the whole thing. So thanks for your, uh, thanks, Peter. Okay, the thank yous. There's some more messages. Uh, 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 okay, going past all that. Uh, with the current improvement in technology, with the mistakes made in the early years of cell in regard to cell death, or I mean, what they were, I sort of hate to go back over it, but we're always sort of, you know, in the sort of blind man and the elephant approach, we're always sort of reaching out, and you don't get anywhere if you don't hypothesize something. And if your hypothesis turns out to be ridiculous, it, that's okay too. At least you suggested you suggested the idea. Is the idea of program cell death get more notice than it's worth? Yeah, sure. But it was an idea that kicked off a lot of sentiment, um, and a lot of activities. If I would just say, and if I were to describe it, one way I've described the position I'm in right now uh, is that I'm sort of a fossil in the field, but it's a sort of special fossil. Uh, you may have, I'm sure you have somewhere in Brazil something similar, but at the top of the Grand Canyon, uh, you walk along, and the National Park Service has helpfully marked uh, areas where there are fossils in the uh, you in the ground there. And the fossils are things like are shellfish, uh, a couple of different kinds of shellfish, and people look at that and say, "Gee, isn't that interesting? How did they get there? But the and then this is a little under 2,000 meters elevation at the top of the Grand Canyon. So, so the question is not really how do they get there, but what that it's clearly this is a mud of an ocean floor of um, the sediment near a shore at the time. And the question is, how did it get to be nearly 2,000 meters in the air? What were the processes that lifted it so high? The processes are everything that has happened since 1964, and they continue with you. Is that there are new ideas that come along, and they keep. So, if anything, I'm just uh, the demonstration that all this work has gone on. And so, were there mistakes? Were there false directions? Of course. Did it matter? No, as long as science, and if you may have noticed in the with the pictures and so forth, they come from all over the world. There are all sorts of people involved in it. I even considered putting a map up of the world of where people who had contributed came from. But that's all things that are for the future of you to do. Okay. Um, that was okay. Uh, um, the new treatments for inducing necroptosis without producing existing. Um, is it important to include the composition of the microenvironment? That's part of the complexity of the whole story that we are, are yet to see. I mean, in my image of the cells dying at different times in culture, one thing that I didn't leave out, and thank you for reminding me of it, is how close are, are they to other cells, and what are the other cells telling them, and or what are the other cells taking from the environment that they could have used. So sure, the a microenvironment is extremely important.
All right, Rick, I think you answered thoroughly all the questions. I think the students have a great starting for this course. And again, we can really thank you enough. It was a brilliant, inspiring talk.
No, you've got it looks like you've got a great course with a lot of wonderful people and wonderful friends that I see. And one thing I will say is that's one of the joys of being in the uh, uh, in the in this field is that during the time that I've been in it um, it never has had the attitude that we used to joke about with the molecular biologists. Uh, uh, as you walking up to somebody in molecular biology and say, "Hi, how are you doing?" So I haven't published that yet. You know, and, the field has, to a very large extent, always been a friendly and open field in which many of the people who were there at the beginning of the field um, remain our friends. And in fact, I have uh, pictures of several of the people in the field in which we have the then and now pictures of when we first met and, uh, and pictures of us today. And we've considered actually going to visit some of these friends now that Zara, my wife, is retired, and so in any case, it's been a wonderful, friendly field in which, with very few exceptions, people have been anxious to share, anxious to learn from others. And if you're going into it, I hope that you have that opportunity that I did.
That's it.
Great, great.
Yes.
Okay, so
Thank you. Thank you. Thank you a lot. It was a amazing
it's been a real
talk.
pleasure. And I
And
hope to get a chance to meet as many of you as I can in person.
Exactly. So hopefully
course.
we will find a way to bring you here and then meet all these young students, the next generation of science, Brazilian scientists. Then
I look forward to it.
yeah, it will be great. So Rick, give our love to Zara and
Okay,
tell them that we miss her a lot as well. And I would tell the students to wait a few minutes after Rick,
okay,
leave and Peter, so we can talk a bit about the course again, okay? Just to explain something else. And tomorrow we will see this other guy, Peter Vandenabili, in the morning.
Yes.
Okay?
okay.
Rick, thank you so much.
Thank
Thank you,
you.
guys.
Bye. Thank you.
Bye-bye.
Okay, bye-bye.